Today we're going to look at something called dihybrid crosses, which is a way to set up a Punnett square to identify the probability of a combination of characteristics to be passed on to the next generation. So for the first example, and actually the only example we're going to cover today, let's look at a parent. Well, first let's draw a key. For the example, I'm going to use the capital R to equal round, and a lowercase r to represent wrinkled. Now, the capital R and the lowercase r are something we call alleles. They are variations of a gene. The capital R, for round, represents a dominant allele, while the lowercase r, for wrinkled, represents a recessive allele. The other characteristic that we're going to look at is for, let's do flower color. And let's use a capital P for purple and a lowercase p for white. Now in this example, I'm going to first say the genotype of each parent, then I'm going to write the genotype of each parent. A good way to remember this is when you're given a problem, you first need to find the genotype. Then you need to FOIL, first outside, inside last, which is a good way to figure out all the possible gametes that the parent could pass on. After you FOIL, you need to finish the Punnett square. Let's look at this example. Let's suppose that we have a homozygous round and heterozygous colored plant. Homozygous round would have two capital R's. Homos is a prefix that means same. And heterozygous purple. And I don't even need to say purple because the capital P for purple is the dominant allele. Now we're going to cross this with a wrinkled, and again we have to use two lowercase alleles. It's the only way that this individual can be wrinkled, and white. And again, the only way that this individual can be white is if they have two lowercase p's. Okay, now what we've done is we've found the genotype. The next step is to FOIL. To FOIL, you first start with the outside. So we're going to start with this first letter, R, capital R, and we're going to pair it with a capital P. The next one, capital R, pairs with a lowercase p. Then we go to the inside. The capital R pairs with a capital P. Capital R pairs with lowercase p. Now what we've done is we've just synthesized something called meiosis. Meiosis is a process in which we take our diploid cell and in essence end up with a finished product of 4 haploid cells or gametes. What we've done here is we have just made or shown the 4 different possibilities of gametes that this parent could pass on to the next generation. We do the same thing for this. We FOIL it out, and what we find is that all the possibilities are lowercase r, lowercase p. Now, let's go ahead and make some room so we can do a Punnett square using the gametes that I just created. Now our Punnett square, since it's looking at 2 characteristics, is going to have 16 squares to it. And what I'm going to do, since I'm going to assume that this is Dad, we're going to go ahead and draw these gametes, place these gametes in the columns above the Punnett square. So capital R, capital P, capital R, lowercase p, capital R, capital P, capital R, lowercase p. Now this lowercase p, lowercase r, lowercase p, is representing mom. And again, we can place this in the corresponding location. Now, to finish the Punnett square, it's very simple. You bring each one of these down and over, and where the letters intersect in the box, you place all of those letters. So each box should contain 4 letters, 2 R's and 2 P's. The only rule that we have here is that we like to list the dominant alleles first, and we like to group the letters together. So for this first example, you would have a capital R. Let me use the eraser here. Alright, so this first example, you would have a capital R, a lowercase r, a capital P, and a lowercase p. Now, since all of the mother's genotypes, lowercase r, lowercase p, are the same, this would be true for the entire column. I want you to write all of them out in your homework and your tests. I'm just doing this to save time. In the next one, you'll have a capital R, lowercase p, lowercase p. And again, that can be contributed to the entire column. In the third column, we have again the same thing as we had in the first column. Basically, this individual is what we would call heterozygous, or a hybrid. And again, it's attributed to the entire list. And the last group is going to be the same as the second column. And that's to the whole list. The only thing that I'm going to ever ask you to do with these is I'm going to ask you to do a genotype percentage or a genotype ratio. Okay, so what I'm going to do is I'm going to go ahead and erase all this over here, and then we're going to go ahead and look at the genotype percentage versus the genotype ratio. If I ask you for the genotype ratio, remember, it's a number in comparison to another number. And if I'm asking you for the phenotype, and by the way, guys, I misspoke a few seconds ago when I said genotype. I meant phenotype, the physical characteristics. That's the only thing I'm going to have you guys find. So the phenotype ratio, when we look at this, we can take, for example, this first one that I'm drawing circles around. That heterozygous individual is going to be round because it has a capital R, and it's going to be purple because it has a capital P. So that's a round purple individual. Now if we count them up, we have one here, and we have one here, and we have one here, and we have one here. That's 4. And then over here in this column, the third column, we have one here, and here, and here, and here, and that's 4 more. That gives us 8 that are round and purple. Now, in addition to that, we're going to look at other phenotypes. The other phenotype that I see, this individual right here has a capital R, lowercase r, but 2 lowercase p's. So this individual's round and white. If we count them up, 1, 2, 3, 4. And we look at the last column, capital R gives this the round characteristic, and 2 lowercase p's tells us that it's white. So these 4 are also round and white. This gives us 8 round / white individuals. Since 8 plus 8 is 16, we know we've gone ahead and accounted for all the individuals here. Now, if I ask you for the ratio, and you've counted up 8 and 8, the ratio is very simple. We would simplify it to 1 to 1. The only thing is that you need to label. round slash purple, and round slash white. That is your ratio. If I ask you for a phenotype percentage, then you simply take the number, for example, the number of round purple flowers, it's 8 of 16, or 1/2. And 1/2 is 50%. So 50% are round purple, and 50% are round white. Just be sure that you label the 50%. And that, my friends, is how simple phenotype percentage is.